Welcome, everyone. This is Cecilia Farah, Marketing Director, Nanion Technologies speaking. And we're very happy to see that you have so many new names in the attendee list. And I would like to wish you all welcome to this webinar. I would also like to welcome and thank today's speakers, Dr. Gerhard Baken from Ionera Technologies in Freiburg, Germany. And after that, Mohammed Kray from Nanion Technologies will present on the Orbit Mini. I will continue with a brief sum up before we move on to the Q&A session. Please remember that you can ask questions at any time throughout the presentations using the chat windows. Make sure to select all panelists so that the application specialist sitting here with me today can see your questions and answer accordingly. Briefly about Nanion. Nanion was founded in 2002 with the introduction of the first planar patch clamp device, the PortaPatch, Nanion has been profitable ever since with the continuous and organic growth of the product portfolio, but also as a company with a loyal and dedicated staff. Headcount today is over 80 people around the globe, from of which 25 are postdoc level electrophysiologists. Nanion has developed a broad product portfolio over the years, not only in the area of automated patch clamp, but also for direct measurements of transporter protein activity or measurements of beating cardiomyocyte networks, where the highlighted products are relevant for bilayer recordings. It's the PortaPatch, the Orbit family, and the Vesicle Prep Pro for automated liposome formation. But in the beginning, there were bilayers. And in this paper from 2001, was published a year before Nanion was founded as a spin-off from the University of Munich and Center of Nanoscience. Here, bilayer recordings from reconstituted gramicidin channels were made on a planar glass chip containing a micro-machined aperture. Here, the driving force to go planar was rather to find new ways to obtain higher bandwidth recordings. And in this setting, it succeeded due to the drastically reduced stray capacitances of the planar glass chip compared to other recording methods. So going planar increased the time resolution and signal-to-noise ratio for bilayer recordings substantially. Another advantage of the chip-based approach to electrophysiology are aspects like automation and multiplexing of patch clamp recordings. So Nanion's first product was the PortaPatch, here on the left. It basically replaces a conventional patch clamp rig and records from one cell or one bilayer at a time. It's easy to learn and use, and increases throughput 5 to 10-fold. And in the following years, Nanion focused on automation and increasing the number of recording channels, and in 2013 reached high throughput screening standards with the introduction of the SynchroPatch 384-768 patch engine, which is capable of 20,000 data points per day with maintained high data quality. So, but bilayers always remained an important and ongoing topic for Nanion, with more and more ion channels becoming available for reconstitution in artificial membranes. Therefore, we introduced the Vesicle Prep Pro for automated formation of giant unilamellar vesicles. So when these burst on the PortaPatch chip, they form a highly resistive, solvent-free bilayer, which then can be used for low noise recordings of reconstituted ion channels. So in 2012, we introduced a completely new platform in collaboration with Ionera for automated bilayer formation and subsequent recordings from 16 individual bilayers. The Orbit 16 simplifies lipid bilayer recordings in that sense that the bilayers are formed automatically at the push of a button, followed by simultaneous recordings from all 16 bilayers, where you just have to add the protein and wait for them to enter the membrane. Data quality is high, allowing recordings also from low conducting channels. And there's a modular approach to the recording substrates. Two different chip types are supported. One module uses borosilicate glass chip substrate, seen here on the left. Basically the same approach as with the porta patch, but with 16 individual recording sites. The bilayers are obtained using giant unilamellar vesicles, bursting over the aperture to form a solvent-free bilayer. The other module supports the micro-electrode cavity array recording substrate developed and provided by Ionera. Here, the bilayer is formed automatically by painting, which Gerhard will explain to you in more detail. And already before turning 1 year old, the Orbit 16 could celebrate its first publication in Science, which was followed by several high-rank publications, reflecting the fact that Orbit 16 is enabling technology, increasing efficiency, and also in the case of Orbit Mini, increasing the experimental versatility of bilayer recordings. The Orbit Mini was introduced a year ago and is a smaller sibling of Orbit 16. It's small, compact, and records from 4 channels in parallel with all electronics integrated, including the 4-channel amplifier. It supports low noise, high bandwidth recording. You'll get the exact numbers on that later from Mohammed. And it uses the MECA 4 chip consumable. What's really unique about this system is that it supports active heating and active cooling, meaning that temperature dependence and temperature activation of reconstituted ion channels can be investigated on the Orbit Mini, as you'll see later. Maybe not technically important, but essential for many research labs and their budgets, is that Orbit Mini is affordable. The complete 4-channel system costs in the range of a high-end 1-channel amplifier. So adding it up, the Orbit platforms allow quick and easy formation of 4 or 16 parallel bilayers simultaneously and the subsequent recordings from these bilayers. It speeds up the experiments drastically with an increased data quality. And additionally, with the Orbit Mini, you can do temperature-controlled recordings. And it doesn't have to be such exciting applications such as porta patch in space or screening 100,000 compounds. We're there to help our customers getting the most out of their assay. And with this, I would like to hand over to Dr. Gerhard Backen, co-founder and CEO of Ionera Technologies in Freiburg, Germany. Previously, Gerhard was scientific co-worker at the Institute of Physiology, University of Freiburg, Germany, where he also received his PhD title in 2008. Gerhard will tell us about the Orbit 16 in his presentation titled Automated Formation of Bilayer Microarrays for Single Molecule Detection with Biological Nanopores. Over to you, Gerhard. Gerhard?
Yeah, thank you, Cecilia, for the nice introduction. And before I start, actually, with the automated bilayer formation, I would like to give you a brief overview of the company Ionera. Ionera was founded in 2014 as a spin-off from the Institute of Physiology at the University of Freiburg. At Ionera, we have developed the MECA technology for the automated formation of bilayer arrays. Ionera is nowadays producing the consumables for the MECA 16 and MECA 4 chips for Nanion's Orbit 16 and the Orbit Mini. The Orbit 16 is an automated bilayer platform. Bilayer can be formed on 16 recording sites in parallel. As previously mentioned by Cecilia, the Orbit 16 features low noise and high bandwidth recording. This platform dramatically speeds up the generation and acquisition of high quality data. As said, the Orbit comes with two modules. Here on the left side, the MECA module, and on the right side, the GUV module. In this webinar, I will focus on the MECA module, which is built for the chip consumable, the MECA 16, which can be seen here. The contact pads on the left and on the right side of the MECA chip are used to connect to the amplifier electronics of the Orbit 16. And the bilayer array is then located at the very center of the chip. In this slide, the center of the chip is shown in magnification here. You can see the 16 bilayer sides, of which each of them is connected and addressed individually to the amplifier electronics, which are integrated in the Orbit 16. On the upper left, you can see a schematic intersection of one of the bilayer sides. It's based on a glass substrate. There are connection wires coming from the amplifiers leading to a microcavity with 6 to 200 micrometers diameter and about 10 to 20 micrometer in height. On top of this cavity, a bilayer is formed in which pore-forming proteins can be reconstituted, and then the current through the protein is measured through an AGCL microelectrode located at the bottom of this cavity. The array size with 16 electrodes so far is giving a large integration density. The 16 electrodes are on half a meter square area. So far, the design is highly flexible, and the geometry of the electrodes as well as of the cavities can be adjusted quickly to any experimental needs. For example, to obtain larger bilayers for low protein concentrations, or small bilayers to even lower the current noise within the measurements. As I will show in the following, parallel recordings are very easily derived, as this technology provides the possibility of automated bilayer generation. Here you see a schematic of how the bilayers are generated. This is a proprietary method patented by Ionera. A drop of lipid solution is placed near the cavities right onto the chip surface simply by dispensing it with a pipette. Afterwards, the lipid solution is distributed by a Teflon-coated magnet on the surface, which finally results due to self-assembling processes in lipid bilayers over the cavity. On this slide, you can see a schematic how the automatic painting is realized within the Orbit 16. The MECA chip is positioned underneath a chamber filled with electrolyte. A Teflon-coated magnet is positioned in this chamber, which can be then moved by a counter magnet underneath the chip, and is subsequently distributing the lipid solution over this little active area marked in orange here in the center, and subsequently forms then bilayers on the chip. But let's have a look how this works practically in the Orbit 16. You can see here, this is the Orbit 16 is a desktop device, and first of all, the MECA chip is loaded onto a little slider, which is then inserted in the MECA module. In a second step, electrolyte solution is added to the chamber above the chip. The wire visible here in the chamber is the common counter electrode positioned from top to the inner of the chamber. On the right side, you can see the current response of all the 16 open microelectrodes to a voltage test pulse of a few millivolts. In a next step, the Teflon-coated magnet is added to the chamber, as well as a first load of lipid solution, which is directly dispensed on the chip surface. Then the magnet is rotated to distribute the lipid on the chip surface. On the right side, you can see how the current response is changing already. Now in a second load of lipid solution, a second load of lipid solution is added, and the magnet is turned at a lower velocity to form bilayers. The current response is now changing towards almost zero here on the right side, indicating the successful formation of a lipid bilayer. Let me show you again how easy that is. You insert the chip into the machine, you fix it, log it in, and then you apply a load of electrolyte into the chamber, followed by the addition of the Teflon-coated magnet, and then you apply first load of lipid solution. We always use 0.1 microliters. The lipid is dissolved in octane. You can also use larger alkanes like decane. Then the lipid is distributed on the chip surface, and finally a second load is added, of lipid solution is added, and then in the second rotation run, the bilayers are formed. Now, let me show to you how the bilayers, the bilayers formed are actually functional. First indication is given by a look at the bilayer for morphology at a microscopic range. What you see here is a single cavity already sealed off by a potential bilayer. We added a little bit of a fluorescent lipid to the solution. You can see the classical annulus has formed at the edge of the cavity and indicated by a higher fluorescence there. And we then applied a voltage of 100 millivolts. As a result, the annulus is a little bit retracted. And finally, at the application of a voltage of 500 millivolt, any fluorescence at the edge of the cavity is gone, and the sealing is destroyed. Again, application of 100 millivolts, classical electrothinning. At 500 millivolts, the bilayer is destroyed. On this slide, images of before here in A and after destroying the potential bilayer are shown. The intensity of the fluorescence is strongly reduced from A to B at the edge of the cavity. The morphology in the magnification is very well corresponding to the bilayers painted in classical setups which are reported in literature. And on the right side, you can see the current response to the change of the applied potential. So when 100 millivolts are applied, you get a tiny change in the current from which you can calculate then the overall bilayer resistivity, which is well above 1 teraohm in this case. Of course, this is the same for all bilayers on the MECA chips. You can destroy them and you can see the reduction of fluorescence. Now, the ultimate proof that the bilayers formed are functional is the successful reconstitution of a pore-forming protein. In this experiment, we used gramicidin as a peptide antibiotic from Bacillus brevis as a stern judge of the bilayer. Gramicidin is only forming pores in functional bilayers of an appropriate thickness. As you can see here on the right side, after the addition of gramicidin, we saw activity on each and every of the 16 channels, and that proves actually that we have formed functional bilayers. Now, one question which is still remaining: how long does it take the lipid solution, which is painted over the cavities, to thin out into functional bilayers? In a classical setup, this usually takes a couple of minutes. So what we did here is we destroyed the bilayers on the array prior to the start of the experiment by a pulse of 500 millivolts, and then reformed them at 0 seconds by one rotation of the Teflon magnet. And as you can see, the activity is restored within seconds on all of the channels. Afterwards, we destroyed and reformed the membranes again and again and again. And again, we had activity in each and every case on each channel. And with the orbit system, you can do this over and over and over and over again. This shows a large advantage of the orbit system. You do not have to set up a new experiment after your bilayers have broken, for example, due to some aggressive ingredient or rough solution exchange. You simply repaint the bilayers within seconds by the push of one button. In the second part of my talk, I would like to show you how the built-in amplifiers in the orbit system perform. The multi-channel amplifiers allow for simultaneous recordings from 16 bilayers in parallel and enable for low noise recordings. For example, at 10 kilohertz final bandwidth, the RMS current noise is below 1 picoamp, or at 1 kilohertz bandwidth, it's lower than 400 femtoamp at a cavity covered with a bilayer. The final bandwidth with the maximum final bandwidth with the built-in amplifiers is 100 kilohertz. With this superior amplifier performance, we were able to reproduce one of the, at least in regards of amplifier performance, most challenging experiments: mass spectrometry of PEG oligomers with biological nanopores. This experiment was reported a couple of years ago by John Kasianowicz, a pioneer in the field of nanopores. A schematic view of the experiment is shown in the upper left. A bacterial toxin, α-hemolysin from Staphylococcus aureus, is reconstituted into a lipid bilayer, and the current through one of those nanopores has a particular size, here indicated by I open. Whenever now a PEG molecule interacts with the pore, a short resistive pulse can be observed in the open pore current, I blockade. Now, this amplitude here of I blockade is dependent on the size of the single molecule interacting with the pore. And now by plotting all these amplitudes into a histogram, you get the size distribution of the analyte at the precision of 1 repeating unit. The detection of these tiny differences in the amplitude has been only possible with high-end single-channel patch clamp amplifiers in the past, but now this is also possible on the Orbit 16 system. Plus, on the orbit 16, you have 16 times redundancy, as you can see from this parallel and simultaneous recording. The 16 graphs here are representing the 16 amplifier channels. And for example, on channel 1 to 5 and channel 7 and 13, 15, a single nanopore is observed. And this not only enables a constant workflow, it also strongly reduces the time to gather high quality data, as the data gathered from one channel can be added up with the others to get a statistically bulletproof results then. And furthermore, statistically rare events, like for example the insertion of low conducting hemolysin pore, like here on channel 8 or here on channel 14, can be revealed. With the MECA chips, it's also possible to go for long-term experiments. What you see here is an experiment where we started with 4 single pore nanopores. The pores on channel 2, 12, and 14 closed within the first 2,000 seconds, and the pore in channel 13 could be recorded for almost 2 hours and was stopped then on intention because the file size reached 1.5 gigabyte on the hard drive. So the Orbit 16 can really add up to your work, which is reflected by a constant flow of publications. Cecilia mentioned it already, in highly ranked journals, of which the first was already published right after the release of the Orbit 16 in 2012. As an example of the experiment which can be conducted with the Orbit 16, I have shown to you single molecule analysis with a biological nanopore, but of course a lot more proteins can be examined with the Orbit 16, like for example the amyloid secretion channel, CSGG. Actually, in our company lab, we have already recorded from a lot of pore-forming proteins. A selection is presented here. The color of the label indicates the way the protein was reconstituted into the membrane. The blue label traces represent water-soluble proteins like antimicrobial peptides or bacterial toxins. The orange label traces are representing membrane-forming proteins inserted into the membrane by detergent-mediated reconstitution. And finally, the green ones represent proteins which were reconstituted by the fusion of proteoliposomes with the lipid bilayer membrane. So all the reconstitution methods known from classical bilayer setups have been tested on the Orbit 16 so far. And if your protein isn't here, we would love to do some test recordings. In summary, Nanion's Orbit 16 in combination with Ionera's MECA 16 chips provides you with the automated formation of 16 stable lipid bilayers at the push of one button. Simultaneous long-term stable recordings are derived from multiple recording sites. The amplifiers integrated in the Orbit 16 enable for low noise and high bandwidth recordings. This greatly reduces the time effort for generating high quality data. Thank you very much for your attention. And I give back to you, Cecilia.
Thank you very much, Gerhard. Excellent presentation. We're going to try to share our presentation here now. Right. Right. So we move on to the next talk by Dr. Mohamed Khia from Nanion Technologies. Mohamed started in electrophysiology at Pierre Fabre before he received his PhD at Jacobs University Bremen. Since 2010, he's application scientist at Nanion and has had a great deal to do with the Orbit Mini and the development thereof. So I hand over to you, Mohammed.
Hello everyone, thank you for being so many attendees. So today I'm going to talk about the investigation of ion channels properties with the Orbit Mini. So first of all, just the overview of my presentation. So I'm going to introduce the Orbit Mini, and then I'm going to talk about the formation of lipid bilayers with the Orbit Mini, which is slightly different from the Orbit 16. And I will finally finish with the investigation of heat and cold sensitive trip channels. So with the heat activating trip V1, trip V3, and the cold activated trip A1 channel. So the Orbit Mini, it's actually several pieces of technology in one device. So it's really a small size instruments as well as a really low weight. It has basically the size of a cell phone, and it has an integrated PLT element and have also a really simplified and efficient Faraday cage. The Orbit Mini exists thanks to the micro electrode cavity array. Compared to the Orbit 16, here for the Orbit Mini, it only possesses 4 wells, but everything is integrated into the chip, including the ground electrode, like I will show in the next slide. And the Orbit Mini has an integrated low noise amplifier with 4 channels that I will also introduce you in the next slides. So you've seen this figure on the left from Gerhard. So basically we have the micro cavity. This has a diameter about 50 micrometers, and below it has a silver chloride microelectrode. So it's filled with solution, and the lipid bilayer is formed on top. On the right side, you can see the chip with the 4 wells and the integrated ground electrode. Everything is so compact, so it allows to have very low noise, and at the same time you can paint all the 4 bilayers totally independently from each other. So inside the Orbit Mini, so you have the eFOUR, it's a 4-channel amplifiers. The great thing with this amplifier is you can record with a very high bandwidth, so it allows recording at 100 kilohertz bandwidth and 200 kilohertz sampling rate. It has a delta sigma architecture. That means that the amplification and digitizer is all in one. It has a capacitive feedback. The eFOUR, the amplifier of the Orbit Mini, is quite different from what is used in conventional patch clamp amplifiers, as it's just bandwidth and signal-to-noise ratio. So there is no additional things that can add some noise to the recordings. That's why the noise figures of the bilayers on the Orbit Mini are quite low, with 800 femtoamp at 10 kilohertz and 7 picoamp at 100 kilohertz, compared to the noise figures of the bilayers on an Axon 200B, for example, which is still the gold standard of patch clamp recordings, especially for single channel recording, where the noise is about 950 femtoamp to 5.5 picoamp at 10 kilohertz, and over 20 picoamp at 100 kilohertz, depending on the bilayer system used. So the Orbit Mini, like I said, it has a PLT element integrated inside, and so you have an add-on, which is the temperature control, which is a separate box that connects to the Orbit Mini and allows to do heat and cold experiments. The PLT element is situated just below the chip, so that you have a control and you have a quite close relation between the temperature set and the temperature that you have inside the chip. The temperature control has a water cooling system, so a heat sink, so that you can basically run the temperature all day long, so with no problems of heating up or having any issue with the temperature. And then you can basically work in physiological temperature with your ion channel or work with thermosensitive channels, either heat or cold. The Orbit Mini has a different temperature range, so you can apply basically from +4 to +55. You have a control through the software of the temperature at all time. And like I said, you can do continuous temperature application. So you have in the graph basically the temperature applied versus the temperature in the chip. And you can see that basically you can apply from -10 to +80 and then have a temperature in chip from 0 to 65. But we recommend to use a temperature range from +4 to 55 degrees, which should be good enough for any kind of experiments. There is a security setting for hot temperature, so there is an automatic switch off in case there is a mistype of temperature. And usually the bilayers are stable within this range of temperature, depending especially on the lipids you use. As you may know that different lipids have different lipid phase transition, so you have to be careful and look at the lipid phase transition of the lipids you use. So compared to the Orbit 16 and MECA 16 chips, so the formation of the lipid bilayer with the Orbit Mini is done by painting. So you have to use a brush or a Teflon brush or a glass rod and then paint your lipid over the wells. It's rather simple to paint. And after painting, so after forming the bilayers, so you can add either purified proteins or protoliposome containing your protein of interest. So usually the protoliposome can basically fuse to the bilayers, and you will see that by changing of capacitance on your bilayer. So once you have the reconstitution of your channels, so you have to be careful of how many channels you want to record. So you can use different concentration and adjust your concentration to record either from several channels, like here, an example with α-hemolysin, where you can see the step of current corresponding to a single channel inserting for each step. Usually what you want to do is single channels, so to better study the open probability of the channels and different basically biophysical characteristic of your channel. For example, here a wild-type KcsA channel. So in the next slide, so the Orbit Mini software is actually called the Element Data Reader, EDR, and allows you to follow everything on the software. So basically the temperature set and applied, if you use the temperature. You have the 4 channels in a single window or 4 windows separately. And the main point is that you can also do online analysis. So you can have histograms of your recordings, as well as dwell time, fast Fourier transformation, or IV graphs right on during the recording. So live experiments and live analysis. You can also export afterwards with the data recorded your analysis and traces. So here are a couple of examples of channels recorded on the Orbit Mini. For example, TSX channel, outer membrane protein channels, potassium channels like KcsA or KV1.3, and we recorded much more channels. If you have any questions about the channels we recorded, or if you have interest on seeing your own channels on the Orbit, just let us know. And now I will start the main investigation that we've done on the Orbit Mini with the heat and cold sensitive TRP channels. So, like I said, the Orbit Mini can allow to do cold active cooling and then record some cold activated channels, or do some active heating and record some heat-activated channels. The temperature range is 4 to 55, and having 4 channels allow us to always have at least one channel, one reconstitution on one of the 4 channels, and then have better statistics every single experiment. So what we did, so we took 3 different transient receptor potential channels. So we took TRPA1 for the cold activated channel, TRP3, and TRP1 for the heat activated channels. And we reconstituted those purified TRP channels into the bilayers formed on the Orbit Mini. So the first channels were TRP1. So we applied different temperature, and we could see an increase of the conductance with the temperature applied, which is totally normal due to the heat. And then we also calculated the open probability versus the temperature. And we plotted this open probability into an Arrhenius plot, and we found, we just calculated the Q10 then, and found a Q10 of 72, and compared this Q10 to the literature, which was basically between 26 to over 100. And we also compared our Q10 results on bilayers with the Q10 that we obtained on the patch liner, so patch clamp experiments done with cells expressing TRP1, and find a Q10 of 82. So after studying TRP1, so we went directly to TRP3 and did basically the same experiments. TRP3 reacted differently to the temperature, like basically an on-off switch with the temperature. So compared to TRP1, where the open probability increased with the temperature applied. So TRP3 had at a certain temperature, which was basically the EC50 at 32 degrees, was basically switching on and having the highest open probability already. We calculated the Q10 from the open probability and found a Q10 of 22 and compared it with the literature where the Q10 was between 17 and 56. We also compared this Q10 with our experiments done on patch clamps or on the patch liner with cells expressing TRP3, where the Q10 was 26. So really close Q10 with the temperature. And we finally used the TRPA1 for checking the cold activated channel. So we actually published in 2014 the TRPA1 results on the porter patch with cold activation and calculated the Q10 previously using the porter patch and found a Q10 of 40 and an EC50 of 14 degrees. So we repeated basically the same experiments on the Orbit Mini, so applying cold temperature and checking the open probability of the channel. And we plotted this open probability versus the temperature and find a Q10 of 46, which was close to our previous results on the porter patch. So as a summary of those experiments and the Orbit Mini, so the Orbit Mini is actually really easy to use for reconstituted of ion channels, and it's really easy to paint over the wells, so there is no tricks. It's just painting lipids over the wells and recording directly. We have really high quality data recording thanks to the amplifier, and so we can basically record very low conductance ion channel, and also use very high bandwidth for detecting fast events. So we can use the heat and cool. So this is enabling the study of thermo-sensitive channels, or simply to work at physiological temperature. And our results with the thermo-TRP channels that we did on the Orbit Mini and defined the Q10 for TRPA1, TRP1, and GV3, and also compare those with the literature and our patch clamp experiments with the patch liner. So determined that it's actually really accurate results, and we got pretty similar results between patch clamp and single channel recordings of purified trip channels. And much more can be done with thermo-sensitive channels and other ion channels reconstituted into lipid bilayers. So to finish, so I'd like, of course, to thank all the Nanion team and the people providing us the protein, Professor Peter Stingman and Lavanya Mopati from Lund University for the TRIPA1 channel, Matthias Winterhalter for Jacobs University for the OMP and the TSX channels, and the company Centralis for the KV 1.3 channels. And finally, 2 weeks ago, Nanion turned 14 years old. So, and yeah, it's great to all those experiments and development of such instruments, and hopefully we'll do much more. And now I hand over to Cecilia.
Thank you very much, Mohammed. Thanks for your presentation. I would like to summarize. I think we've all seen here today that the systems are enabling and easy to use. The increased throughput due to the parallelity of the systems using either 16 or 14 recording channels in parallel, providing high data quality and also unique features such as temperature control. And finally, Nanion has a large installed base of products around the globe, where more than half of our customers are academic scientists, which also is reflected in the ever-increasing number of publications where our products have been employed. So with that, I would like to move on to the Q&A session. We're receiving many questions today, so the ones that we're not answering now in this session, we will answer per email, and we'll also send out a document with questions and also providing the answers. Okay, so let's start. Gerhard, are you with us?
Yes, I'm with you.
Great, great, because I have one question here for you, also for Mohammed. It's a question regarding signal resolution of both the Orbit 16 and the Orbit Mini. But regarding the Orbit 16, what is the capacitance of the system once you have a bilayer? And what are the corresponding noise levels during the experiment?
So the capacitance of the Orbit system is about 25 picofarad with a bilayer-coated cavity. And the corresponding RMS noise levels during an experiment are below 1 picoamp at 10 kilohertz final bandwidth, or below 400 femtoamp at 1 kilohertz bandwidth. This is very important to resolve small pore-forming proteins like e.g. ryanodine receptors or KcsA at physiological conditions. This has been done before, but with a classical bilayer setup. But here, one of the strengths of the Orbit 16 comes into play. You can record those channels at higher bandwidth, and so see about, for example, their gating behavior more clearly. And that is because the overall system's capacitance is rather low.
All right, thank you very much. Same question to you, Mohammed. What's the capacitance of the system once you have a bilayer for the Orbit Mini, and what are the corresponding noise levels during an experiment, a typical experiment?
So That's a good question. So the capacitance of the Orbit Mini with the MECA 4 chips is around 10 picofaraday with a bilayer using DPHPC lipids. The capacitance is really small as everything is actually tightly put together on the Orbit Mini. The noise level for the bilayer is around 800 femtoamp at 10 kilohertz. Like I said in the presentation, this low noise allows us to measure really low conductance channels or channels like chloride channels, for example, with as low as 5 picosiemens, 10 picosiemens conductance.
All right, thank you. And then a question, both on Orbit 16 first, but also Orbit Mini. Can you adjust the voltage protocol for every well individually? Mohammed, could you address this?
So when using the Orbit 16 with the Tesla amplifier, it is possible to apply a voltage protocol to individual channels, having basically 16 different possibilities. But when you're using the element amplifier, for example, on the Orbit Mini or the Orbit 16, so only one voltage protocol is possible to use on the 4 or the 16 bilayers.
All right. Then a question about data analysis. Can you do data analysis in separate software packages? That is, can you easily export data, or is it rather a closed system? Mohammed.
Okay, that's also a good question. You can do the analysis in a separate software package. The recordings are saved in different formats, including ABF, DAD files, or CAV files, so you can basically open the traces and do the analysis on, for example, ClumpFit or Origin or even Excel as you wish. So it is really possible. We also have online analysis that you can basically save into the format, so you directly have the analysis of your recording in different format as well, so you can save the online analysis into CSV files and then having directly into Excel, for example, and without doing any extra work on the analysis. So you directly have the ready to go for publication data.
Sounds good. Gerhard, there's a question to you regarding the MECA chip consumables. Can MECA chip consumables be cleaned and reused for several experiments?
Yes, they can. They can be reduced to a certain extent. It very much depends on the frequency of washing of the chips, as they have a certain amount of silver chloride storage there. So the more you wash, the shorter the usage of the chip is. And it also depends on the current levels measured. But they hold for at least 2 days. However, we strongly recommend to change the chips if you switch the protein you put on there to avoid any cross-contamination.
Okay, I see. Another question regarding the MECA chip consumable. What are the different cavity sizes, and can these be customized?
So our standard cavity sizes are 16 micrometer in diameter and 50 micrometer in diameter. And yes, we can customize the diameter. We can also, to a certain extent, 15 to 25 micrometer, customize the height of the cavities. The range for the diameter, I mentioned it in my talk, is 6 to 200 micrometers. which can be customized.
Okay, thank you very much, Gerhard. So I think we end here, and as already mentioned, all questions will be answered in a separate email after this webinar. So once again, thanks so much for attending today, and of course a big thanks to Gerhard and Mohammed for your excellent talks. And as already mentioned, if you have questions, if you would like a demo, if you would like to see the systems in action, just let us know. We are happy to help and to show your system, to show the Orbit platforms in your lab. So with that, I would like to say goodbye and thanks for joining.